Assalamu alaikum. First of all, I want to thank Professor Shahid Parvez and CPSP for arranging this educational activity for chemical pathology. In the time of epidemic, such activities are very useful for the trainees and for the junior doctors. So the topic allotted to me today is electrophoretic techniques. Electrophoresis is a very important analytical technique which we use for estimation of various laboratory analytes. In this talk of about 15 to 20 minutes, I will very briefly go through these techniques. This is the my learning specific learning outcome. Now, as usual, I will ask some questions, and if there are some trainees attending this, uh, the seminar, please write the answers with yourself, and so that you remain active throughout the talk. So, what is a common feature of electrophoresis, chromatography, and mass spectrometry? What is common in these three: electrophoresis, chromatography, and mass spectrometry? So, common feature: all these techniques are used for separation of substances in a mixture. Basically, all these techniques are separation techniques. But in addition to separation, we also quantify the substances, and then we can use it for diagnosis of various diseases. So, what is the difference? What are the differences between electrophoresis and chromatography? Let's see. Rather, you write on your paper what are the differences between electrophoresis and chromatography. Electrophoresis is basically the electrical in electrophoresis. I'm sorry. For in electrophoresis, we use electrical property of the substances for separation. Whereas in chromatography, there may be electrical activities which is used for the separation of substances, but other properties are also used. For example, physical properties, chemical properties. Here we call them partition coefficient. So by partition coefficient, we mean that the characteristic of the substance which is being used in chromatography for separation. It can be electrical charge on the substance. It can be physical characteristics. It can be chemical. So in chromatography can be varied in partition coefficient. There are essentially a stationary and mobile phase in chromatography, whereas in electrophoresis there is no stationary and mobile phase. Electrophoresis and mass spectrometry. What are the difference between these two? Mass spectrometry is also an electrical base separation technique. Rather, it is based on the charge to mass ratio. But uh, for electrophoresis, it is essential that the substance which we are going to measure has charge on it. Uh, proteins have charge on it, so you can measure it. But in mass spectrometry, if the charge is not present on the substance, the charge is produced. How charge is produced? By ionization. So, mass spectrometry, ionization is a very important part. By ionization, bombardment of the electron on the substance, ions are produced, and then those ions or electrical property can be used for separation, whereas no such thing, ionization is not present in electrophoresis. Sometimes rather, it is very common now, mass spectrometry fragmentation is also done. That means the fragments, this compound is broken into fragments and then ionized. No fragmentation is carried out in electrophoresis. Now, what are the types of electrophoresis? I will ask the questions, and I'm sorry for those students who already have gone through these type of my talks, but I think they will also uh, uh, take it as a revision. So my first question is, I will write. I've written a description, and you will tell me which technique it is. Proteins and other molecules move to cathode or anode depending on charge on the surface, but at a very specific pH. Proteins and other molecules become neutral and stop migration. Ordinary diffusion is also counteracted. These characteristics are indicative of which electrophoresis technique. So, what do you think it is? Which type of electrophoresis in which the the molecules, the proteins especially, they become neutral and then stop migration? Yes, it is isoelectric focusing. So, what is the principle of isoelectric focusing? Before going to that isoelectric focusing, let me go to a very basic thing, that proteins are amphoteric compounds. You might have read in the basic biochemistry that proteins are amphoteric. That means it can be positive, it can be negative. By wherever the word amph emph is there, it means having two properties. When we say this vehicle is amphibian vehicle, it means it can move on the land as well as on the sea. Similarly, amphibian animals are those which can live on land and the water at the same time. So, amphoteric compounds or proteins are amphoteric because their charge depends on the pH of the buffer. At certain pH, it becomes positive, and in other pH, it becomes negative. So, but there is another very interesting concept of proteins, which is related to their amphoteric nature, and this is called isoelectric point. Isoelectric points are those points at which the charge on the protein becomes zero. The protein no more remains negative or positive. So if we move these proteins through their specific isoelectric points, their movement will stop, and then we can identify that this is that particular protein because this specific point is for that protein. So by employing isoelectric points, we can identify, quantify uh, the proteins, and the actually by pH gradient, it forces the protein to remain in this uh, isoelectric point, and this concentration effect is called focusing, because protein that particular protein that concentrates on a particular point, so that is called isoelectric focusing. Second question is electrophoretic separation based on mass to charge ratio and molecular size of the compound, resulting in improvement in resolution, for example, separation of proteins. What is this technique? Yes, it is polyacrylamide gel, PAGE. Polyacrylamide gel is a very popular technique, especially in the world of genetics, because polyacrylamide gel it can we can use it uh, very thermostable, transparent, strong, and relatively inert gel, which can be made in a wide range of pore sizes. Separation is based on both charge to mass ratio and molecular size, which is also called molecular sieving. There is no endosmosis. What is endosmosis? It is the movement of the medium, and we will talk about it a bit later. We have precast, that means pre-manufactured mini gels available in a variety of concentration for most protein and nucleic acid. It's very interesting to tell you that PAGE is a very popular technique for nucleic acid separation. Uh, Prior to the real-time PCRs, the nucleic RNA and DNAs, they were identified by using this PAGE technique. Still, if some nucleic acid has to be used in a research and no real-time assays available, PAGE is used. So very popular technique in genetic analysis. This is a diagrammatic representation of PAGE. You can place these are the uh, this is the medium and the buffer, and we can separate the large high molecular weight proteins from low molecular weight proteins, just because of their not only the electrophoretic ability but also the pore size. So that's why, because of these dual type of separation characteristics, we can separate, or we can increase the resolution of protein separation. Thank you. Proteins are stacked before separation, and there is no need to carry out step of pre-concentration or specimen with low proteins. Resolution is much improved. So, what technique comes in your mind? Those who have not understood the question, let me explain it a bit. Sometimes the concentration of protein is very less in a solution, protein or any substance. It is very less, a very small concentration is present. So we have to do a step, extra step before electrophoresis, which is called pre-concentration step. That means the concentration of the of the substance in this specimen is increased. But then there was a technique developed. A technique has been developed in which the this pre-concentration step is dispensed with. It is no more required, and this technique is called disc electrophoresis. And by disc here means discontinuous electrophoresis. Why it is called discontinuous electrophoresis? Because it uses two gels, and these two gels have got different pHs. Uh, because of different pHs, the protein which move between these two gels, they are separated in very sharp bands. So we can have a very high resolution, as well as we do not require a pre-concentration steps. It can be used in for those biological fluids in which the quantity of protein is usually very less, for such as CSF. So this is a diagrammatic representation. This is gel number one, and this is gel number two, and the this is the bridge, and the protein, when they move from one gel to another, they get separated, and we can get a very sharp band. This is called discontinuous or disc electrophoresis. Number four is high resolution separation of charged substances without a sieving matrix and with application of high voltage, small sample volume, and short running time, ease of automation, and extensive applications. I hope you might have reached what we are talking about. This is capillary electrophoresis, which is also called capillary zone electrophoresis. Capillary electrophoresis is very popular these days because it has made the lives of laboratory staff very easy, because it is very user friendly, its throughput is very high, and the results are very good. So, mostly in labs now, you will find capillary electrophoresis instead of the traditional electrophoresis. In capillary electrophoresis, there is a very small bore uh, fused silica capillary tube, which is coated with thin polymeric covering its exterior. Inner surface carries negative charge. On the inner surface, there is negative charge. A ionic cloud is also created. When we add a substance, it creates an ionic cloud, which is predominantly positive charge. So as this uh, electroosmotic flow is main driving force, all sample components migrate towards the negative electrode. Now you can see here that this is the inlet, and we put the sample here, and it moves from positive to the negative charge. So logically, which ion should lead this migration? Of course, the positive ion, cations. Cations will be ahead of the all the ions, followed by neutral ion, and then negative ion, because it is moving from the positive to the negative charge. Here you can see that cations, they move the fastest, followed by neutral ions, and then anions. So in capillary electrophoresis, we have this characteristic that uh, positive ions, they move fastest. Second point is that The total mobility in capillary electrophoresis, which is called V tot, is the sum of two mobilities. One is electrophoretic mobility, which is like other in traditional electrophoresis, and there is also electroosmotic or endosmosis mobility. This is a new thing. This is a unique thing for capillary electrophoresis, that it is not present in the traditional electrophoresis. So endosmosis plays a very important role for increasing the mobility of the substances in capillary electrophoresis. First, I will show you this slide. Uh, if you remember, the point of application in traditional electrophoresis is cathode, negative electrode. So the movement is from cathode to anode, and the protein which leads in the mixture is albumin. The highest peak is of albumin. So in traditional electrophoresis, point of uh, application is cathode, and movement is from cathode to anode, and the front is albumin, followed by immunoglobulins. But in capillary electrophoresis, it is reverse. The point of application is anode, and the migration is from anode to cathode, and the leading front is immunoglobulins. The immunoglobulins they lead, and albumin they lag behind. So these are very important two differences between the uh, capillary electrophoresis and traditional electrophoresis. So, what are the advantages of capillary electrophoresis? Simple with selective separation mechanism, efficient heat. Because very high voltage is applied, a lot of heat is produced, but a very efficient heat dissipation mechanism is present, which prevents denaturing of the protein. Uh, so enhances separation efficiency. The turnaround time is very less. High degree of system automation. It can be automated, and it can be interfaced with your computer system online, and so that is very good. And the actual sample need is nanoliters, but of course you can add a few microliter in the capillary. What are the applications of capillary electrophoresis? It can be used for serum, urine, CSF, body fluids. It can be used for hemoglobin variant lipoproteins, carbohydrate deficient transferrin, and many others. So, capillary electrophoresis is now widely used because of its high throughput, its accuracy of results, user friendliness. Only thing is that it is very costly. Thank you very much.